Good morning and welcome to our GEOTP-A seminar. And this morning we have Mariel Vazquez, who's going to talk on topological modeling of DNA recombination. Mariel.
Thank you, DeWitt. So first I want to thank, all of you to be here. If you're in the West Coast like me, it's very early for you, so thank you. And I want to thank the scientific committee of the GEOTP-A seminar series for the invitation to give this talk. And today I'll tell you about my work on topological modeling of DNA recombination. And this is a topic that has kept me very busy for many, many years. And a lot of different sub-lines of research have come out of this topic. So I will touch on some of that. It has paved the way for many other projects, and it has provided a lot of opportunities for training. So I am, as you see on the title slide, a professor in the Department of Mathematics at UC Davis and also in the Department of Microbiology and Molecular Genetics. I have a relatively large group that I co-lead with Javier Arzuaga, and here's the picture, the most recent picture of the group. The people at the bottom right in the inserts are either people who couldn't be at the photo or who left recently. So Michelle and Allison left recently, but I'm going to talk about all of their work, so I wanted them to show up here. And in this crowd, I mean, you see everyone from undergraduate students to graduate students, postdocs, and they represent many different disciplines. And this is what our group does. We put a lot of different disciplines combined with mathematics to understand problems in biology. I also wanted to acknowledge on these first slides the grant funding that allows me to do this research and to fund some of these people. And because I am talking about work that has been going on for many, many years, I also want to show you a list of acknowledgements because, of course, this work is not done overnight and it's not done by one single person. So there's many people to thank over the years. And here's a long list for this talk, and I'm assuming you see my mouse. For this talk, I will be talking about the work of Alison Moore, who was a Krenner assistant professor here at Davis. That's like a temporary non-tenure track assistant professorship that is a fancy postdoc if you wish. And she spent in Davis 3 years. Now she's an assistant professor at Virginia Commonwealth University, so I should change that affiliation. And Alison's collaborator, Ty Littman, we did some work together. And then there's many other people, in particular, of course, the Whit Sumner should be at the center of this acknowledgement page because everything started with him, but he's here, top center. And then Koya Shimokawa, Kai Ishihara are low-dimensional topologists in Japan. They played a big role in this work. And then the experimentalists, David Sherratt and Ian Grange in Oxford, and some people who were fully devoted to the computational aspect, in particular Rob Sharine, who does, a lot of you know, because he is in charge of Notplot. He created Notplot. And then many other people in my group throughout the years, students and postdocs. All right, so GEOTP-A stands for Applications of Geometry and Topology, and that's really at the heart of what we do. And something beautiful about this series is that there's many ways to apply geometry and topology, and ours is just one flavor. So let me show you what that is. The outline of the talk is as follows. I will motivate the problem with a very specific question in biology. And that question is, how does DNA topology get simplified during DNA replication? If you're not familiar with the biological terms, I will explain everything, and I always try to explain things with pictures, but you can always turn your mic on and stop me and ask me questions. Then we will talk about a mathematical model of local reconnection. That will take us to discussing pathways of unlinking by local reconnection. We will do that discussion both from an analytical perspective and a computational perspective, where I will show you the computational tool that we use and that we developed in our group. And at the end, all of this requires in the back of your head to be thinking about the importance of chirality. So I will tell you about what we've done recently in that sense. And I hope I won't run out of time. All right, so motivation, topology, simplification after DNA replication. These are the three key players, but there's many other people, and there's 3 more people listed here, and then there's many students involved in this project. What is DNA replication? On the left here, you have the traditional representation of the double-stranded DNA of a double-stranded DNA molecule that consists of 2 sugar phosphate backbones. Those are the blue ribbons, and they come together connected via hydrogen bonds, nucleotide with nucleotide. So A with T and C with G, and those are color-coded here in the center, like the steps in a staircase, in a winding staircase. And during DNA replication, enzymes, which are big proteins that bind to the DNA and do something to the DNA, come together, they bind to the DNA molecule, they break the hydrogen bonds and unwind the helix. That's represented here by this triangle. The region where you see the triangle is called the replication fork. In the center, this is an imaginary line, which is representing the axis of the double helix. When we're talking about winding of DNA, in general, we will forget about the double helix itself, and we will just think about what the axis is doing. So we usually like to represent the axis in our images. And on the right, you see the axis right here in purple. As the replication fork proceeds, in this picture, it would proceed from bottom to top, and the double helix is being unwound. If you imagine that the very top is fixed, this unwinding of the helix is causing a tremendous amount of torsional stress right here in this region where my mouse is. And because DNA is a biopolymer that likes to have a certain helical pitch, it likes to have 10.5 base pairs per turn of the helix, no more, no less. Any overwinding of the helix will result into what biologists call supercoiling of the molecule around itself. So that means coiling of the axis, and you can see it on the right side, around itself. So this is something that is happening every time the DNA is replicating. Why does a cell need to replicate DNA? Well, every cell has a genome that is composed of 1 or more DNA molecules, and this genome needs to be faithfully copied before the cell can divide into 2 cells. The process of copying the genome is called DNA replication. So this is happening every time the cell is going to divide, and before dividing, you need to get 2 identical copies of the original DNA molecule. All right, and every time you copy the DNA, you have this overwinding and this geometrical conundrum that is depicted on the right. Now, if you look at this picture for a second and you think that the DNA molecule, instead of being a linear piece of DNA, as you see here on the left, instead is a circle, then we can start interpreting this in a different way. If you have a linear piece of DNA, the torsional stress, maybe locally, the DNA will wind upon itself as in the right, but in general, that winding, that torsional stress will fall off the end of the molecule because the molecule is open and linear. Now, if instead of starting with a linear DNA, you start with a circular DNA, now this winding will stay there. It will not fall off the end. So in the next slide, we have a little cartoon. The black oval represents a cell, your favorite cell. In general, if we talk about circular DNA, we're thinking bacteria or archaea. and bacteria in particular have one single circular chromosome. So that's depicted here in green. Ignore the fork right here. So just imagine a green circle. The green circle has a black dot. That black dot is the origin of replication. And then in this picture, we see two replication forks. So the forks are indicating to you that replication has started. It happens bidirectionally. You have one replication fork right here moving from left to right toward the blue arrow, and one down here moving from left to right toward the blue arrow. Now, if you remember what I said in the previous slide, the DNA is a helix, so each replication fork will cause the DNA to overwind ahead of the replication fork. So here is a more detailed, still a cartoon, but a more detailed image of that showing the double helix right here and the replication forks moving. And as you proceed from left to right, you will have supercoiling ahead of the two replication forks. There is a way to assign a sign to the supercoiling. This will be what we call an accumulation of positive supercoils ahead of the fork. This process is moving very quickly. The torsional stress is very high, so it is believed, and there's been some experimental evidence, that some of that torsional stress will diffuse behind the forks and form what biologists call precatenanes, that mathematicians would call pre-links. So this is something that already looks like a topological link, but there's these forked structures at two locations. What will happen at the end of the replication? At the end of the replication, you expect these forks to be resolved and the pre-link to become an actual link. And here is an image of the some of the experimental evidence for this type of structure. Here you have a circle of DNA that is in the process of being replicated. The two dots represent the two replication forks. Here you have the accumulation of positive supercoil ahead of the replication forks, and here you have the pre-links or precatenanes behind the forks. And this is a line drawing of that image. The image comes from the electron microscope. It's an electron micrograph of actual DNA. And this was published in the late 1990s. Okay, so at the end of replication, if we recapitulate, we start with a single DNA circle, which may be supercoiling. So here now, I'm only drawing the axis of the helix for you. We're not looking at the winding, at the coiling of the DNA around itself in the sense of the two sugar phosphate backbones forming the double helix. We erase the double helix. The double helix is that yellow tube, and it may be supercoiled. DNA replication goes through, as we showed in the previous slide, and at the end of replication, we have an accumulation of two component links. For the topologists in the audience, these two component links have been characterized experimentally, and they're all right-handed torus links of the form T, T parenthesis 2, 2M. And so here, for example, we have the 4-crossing link, the 6-crossing link, the 8-crossing link. You can also have the Hopf link, which is which only has two crossings, or the unlink, and conformations with much higher crossing number. Now, okay, let's remember why the cell needs to replicate its DNA. The cell needs to replicate the DNA, to copy the DNA, so that eventually, when the cell is ready to divide into two new cells, each new cell inherits one identical copy of the genome. Well, but now your two identical copies of the genome that need to be segregated into two new cells are interlinked. So we have a topological problem here. And let me emphasize that this is not the product of our imagination. This is happening all the time. If every time a circular DNA molecule tries to replicate, there will be this accumulation of torsional stress, and you are expected to see these links forming. Now, of course, this is a problem. The cells cannot divide and each cell inherit one of the molecules. Something needs to happen, and what happens is that enzymes called type 2 topoisomerases come in, and they act like topological scissors and molecular scissors. They go in and then they undo the crossings. They unlink the linked DNA to get you back to two identical copies of the genomes that now can segregate at cell division. Okay. What takes me to my topic, and I'm also very interested in studying topoisomerases. We've done some work throughout the years, but today I'm not telling you about that. I am telling you about what happens if the topoisomerase doesn't work. Now, topoisomerases are enzymes that are essential to life, and they're ubiquitous, meaning that they have been found in every organism that has been studied. They have been found, they're there, and if you get rid of them, the cells typically die. A lot of antibiotic drugs and some anti-cancer drugs target topoisomerases for that same reason. If you're infected with a bacterial infection and you take a drug like ciprofloxacin, for example, that drug will inhibit the topoisomerase, and then the cells, either for topological reasons or for other reasons, will die, and eventually you get better from the infection and your body heals. But we all know that there is a lot of bacteria that are resistant to antibiotics, and in particular, bacteria. There's a big class of bacteria, a growing class of bacteria that are resistant to antibiotics that target the topoisomerases. So a question that is always in the minds of people dealing with drug design and people in the medical fields is what is causing that bacterial resistance. And if the topoisomerase in particular is not working, the cell should die. Why are the cells not dying? Why are the cells recovering? Maybe the cells develop a way to not absorb the drug, and therefore the topoisomerases are not inhibited, and therefore the cells don't die. But maybe the drug did come into the cell, and somehow some other process recovered the cell. So that's what we were thinking when we started this project. So we were thinking about how do we model topoisomerases in the first place. And if we think about topoisomerases, they are, of course, and they sound very sophisticated, molecular scissors that will unlink the DNA, but their action is very simple and naive. Their action is binding to one crossing in your link and just performing a strand passage. That's the terminology in biology. In mathematics, we would say that topoisomerases act by crossing change. They just introduce a crossing change, and remember, each one of these strands in the crossing represents the axis of the double helix. Now, the Sherrod lab in Oxford showed a few years ago, so a little bit over a decade ago, that when the topoisomerases are not working, there's another system in the bacteria E. coli that can unlink the DNA links that come as a result of replication. That system is called XerCD, the FTSK. It's a complicated name, but if I break it down for you, XerCD are other type of molecular scissors. They're called site-specific recombinases, and they act by binding two strings of the DNA, introducing two breaks. The topoisomerase only introduce one, so here you introduce two breaks and reconnect. So the site-specific recombinases bind these two pieces of DNA. The pieces of DNA in this particular experiment are called diff. So diff represent the two pieces of DNA, and FTSK is another enzyme that is required to see efficiency in this reaction. It's a translocase that is believed to help with bringing the two pieces of DNA together. So that breaks down the name for you. But the point is that they remove the links, and if we look at them in a cartoon fashion, again, each one of these blue and red arcs represent the axis of the helix, and these are very short pieces of DNA. Introduce one break in each, reconnect, and reseal the breaks. Mathematically, this reconnection, and I will refer to it a lot as local reconnection, is modeled by a band surgery operation. I will talk more about that later. And if you're curious how things look in a slightly more realistic fashion, here is your DNA in yellow, and these are the proteins binding to the DNA in purple and green. Still a cartoon representation, but it gives you a glimpse of the complexity of the binding. But one thing to point out is that the piece of DNA that is being bound is very small. What do the experimental data look like? This is the type of data that we obtained from our collaborators, and what they did is they generated these links after replication. You see them on the very left column, and if you're a mathematician, most of you are probably mathematicians, you have no idea how to read this, and it's complicated. And yes, it is complicated. It takes time to learn to read these gels, but all that I want to tell you is that this is a time course. The time is here, 0 minutes, 2, 5, 10, 30. This is a control column. Different temperatures, 25 degrees, 37 degrees. And for each time point that the experimentalists chose, they incubated the DNA after the replication process. So first the DNA underwent replication. The links were purified and taken out at time 0, you see the links right here. Each of these bands represent a topology. So the top band will be the Hopf link with 2 crossings. This band right here is a 4 crossing link, 6 crossing link, 8 crossing link, 10, 12, 14, and then the unlinks migrate separately at the bottom. At 2 minutes, you're incubating those links with the enzymes, with the molecular scissors, with XerCD. And slowly over time, we see that the number of unlinks increases and the number of complicated links decreases. And it's not a super clean experiment. It's hard to interpret. It's hard to be sure what you see here. So we set out to try to understand what the enzymes were doing. And one thing to point out is that there's intermediate bands, and they're darker and darker as you move in time. These intermediate bands were confirmed in another set of experiments to be representing knots. So here you have 2 component links on the left, and the intermediate bands are single component knots. Okay, so these type of experiments led the experimentalists to propose a model to say, okay, if I have a replication link, I know that it looks like the one here on the left. It's a 6 crossing torus link that mathematically we write as T2,6. The P represents the orientation of the recombination sites, and orientation will be important. I'll mention that later. And we know that this link has to be unlinked. The question is, how does it do it? What is happening at every step of the reaction? How many steps do we need to go from the 6 crossing link to the unlink? And the biologists came up with this model where they said, well, unlinking is happening stepwise, where you go from a 6 crossing torus link to a 5 crossing torus knot to a 4 crossing torus link, 3 crossing torus knot, the trefoil, 2 crossing torus link, the Hopf link, on knot, unlink. So a very simple mechanism, very easy to understand. So what are our questions? Is this really the topological mechanism? And then for each arrow in the previous picture, can we understand exactly what is happening? What is the topological mechanism of unlinking? What intermediate link types arise during unlinking of a substrate link of type L? So in the previous picture, we had a 6 crossing torus link, but it could be an 8 crossing, a 10 crossing. It could be some other type of link. So we're interested in the general question from a topological point of view. What if you have any other type of link, like a Whitehead link or some other type of link? What intermediate link types do you see if you unlink by performing local reconnection? And then once you see once you start with a link and you're going toward the unlink or toward another link, is there only one way to do that, or are there different pathways to do that? So those are the 3 key questions. And here I want to make a detour because this this series is focusing on applications. So you've seen talks by many different people with applications, not only to biology, but also to physics. And examples of local reconnection happen in many different settings, not only with DNA. So different settings and at different scales. We saw the DNA case where we have it at the microscopic scale. And let me point out that the same questions apply in any other scenario. The answers are independent of the setting, unless I tell you this assumption is a biological assumption, but the analytical tools will not change. The assumptions might change based on the problem. And the methods we propose should work for any of the other problems. So at the microscopic scale, you have DNA. At the macroscopic scale, you have magnetic reconnection in solar coronal loops, and the reconnection looks very, very similar to what you see in DNA. You bring two of these loops together, and then there's a reconnection event happening right here, and at the bottom you see the result of the reconnection. Then at the scale where we live in, we can look at knotted vortices, fluid vortices, and if I have time, I'll tell you a little bit about these experiments, but these were experiments done at the University of Chicago in the Irvine lab, and in these experiments, the experimentalist, the physicist, blew some water flow through a 3D printed hydrofoil, and from this experiment resulted a fluid vortex that could be unknotted, like in the top here, or knotted or interlinked, and I'll show you a movie of a linked one later on. And as this fluid vortex dissipates over time, it's very clear from these experiments that reconnection happens right here, right here. And the reconnection looks very, very similar to what we see for DNA. So our goal is to understand the topological mechanism of local reconnection using geometry and topology. This is about half of my talk, so maybe people have questions before I go into, although it was a preamble, but if there's questions, this is a good time before I go into the model. No? Okay.
Mariel, if I may, yeah. I have a question.
Yes.
I'm just curious you mentioned this second mechanism, you call it XerCD. Well, comparing this mechanism with the typical type 2 topoisomerase mechanism to unlink, which one would be more energetically expensive? I believe probably the XER.
Yes. The topoisomerases are very efficient. So with strand passage, if you think of the torus link that we saw, so this picture right here, with recombination or reconnection, if you do a reconnection here on the left, in this particular scenario, it takes you from a 6 crossing to a 5 crossing. If instead of reconnection, you had done strand passage, you would, for example, change this crossing right here, and you would jump directly to the 4 crossing. And then from the 4 crossing directly to the 2 crossing. You're not going through knotted intermediates. You're not changing the number of components. You're literally going with scissors and just unweaving. And you can, the topoisomerases do not choose one of the colors, blue or yellow, but if you were doing it yourself with scissors, you could just choose to cut to damage, if you want, the blue one and leave the other one intact. In recombination, the number of steps more or less doubles. This is not exactly right what I'm saying, but for this particular example, it is correct. The number of steps doubles, and it won't be as energetically favorable. Topoisomerases are very efficient and very, very fast. But if the topoisomerase is treated with a drug that inhibits it, which was a case of these experiments, the question is there another process that is rescuing the unlinking? Does that answer your question?
Yeah, certainly. Can you quantify how much more energetically would be the task for XerCD?
Well, I guess you could quantify it. We're not really quantifying energy in this work, but you could assign energies, and depending on what energy you're looking at, yes, of course, you could quantify it, especially when we go to the computational experiments. You could think on how to do that there. And I mean, we are doing some things that involve worm-like chains, and there's energies involved, but I'm not reporting on that yet. Okay, so the mathematical, yes? Yes.
Sorry to interrupt, but I've got a question. How does the speed of reconnection compare to the speed of strand passage?
The actual speed in seconds, I don't know. Yeah, I don't know. That's a good question. Yeah, and I don't know. I mean, just from my own experience, but this is not to say that that's the speed. But the experiments that we did with Joaquim Roca on the unknotting of those phage knots, that time course, in a matter of seconds, all the knots are gone. Here it's minutes, and the links are not all gone. So that suggests that this is much slower, but I don't know if that's true, or I'm just being misled by the small sample size. Yeah. Okay, so the mathematical model. So again, let's remember, I think I wanted to move this slide further, but well, we can remember this slide. So in a cartoon fashion, local reconnection acts by bringing two strings together, two short pieces of DNA together. So each one of these, again, represents a double helix, but a short amount of DNA. They come together. Here we call these zero tangle with parallel orientation. So the orientation of the DNA will be important. And this is not the chemical orientation of DNA. This is an orientation that is imposed by the model based on the sequence. These two strings have the exact same sequence. If you're reading from left to right and you read the same word, then you have parallel orientation. If you read from left to right and you read the opposite word on the bottom than in the top, I mean the reverse, if you wish, then you have anti-parallel orientation. And if the two strings are horizontal like that, we call that the zero tangle. Reconnection goes by a band surgery, and I'll give you the formal definition a bit later, but basically you're cutting the top segment of DNA, the bottom segment of DNA, and you are reconnecting. In the case of parallel, the reconnection will take you to adding a crossing, either positive or negative. The one shown here is a negative crossing. In the case of anti-parallel orientation, reconnection, because you want the orientations to be what we call coherent, you want to go head to tail and head to tail, then reconnection in this case will take you to two vertical strings, no crossings added, and R is called 0, 0, or the infinity tangle. Okay, so reconnection between two sides along circular chains, and this could be knots or links, so your substrate is either a knot or a link, and I already convinced you that they can change the topology of the chain. We saw the example of replication, and the reconnection is a black box. We want to understand all these different things that I asked in my questions slide. Mechanism and pathways and shortest pathways. It's been known for many years that reconnection, and in the case of DNA, is in the form of site-specific recombination, can change the topology of DNA. So that's our key example, and what made all of this start for me, and I think for the Whitsamners. Site-specific recombinases cannot unlink circular DNA, and these are two traditional examples, the lambda integrase from bacteriophage lambda, and this XerCD that started being studied in the 1980s. Same for both of these. I mean, the topological effect of these enzymes was studied for the first time in the 1980s. And you can see some of the old papers right there. But these two enzymes were observed to tie knots or links in the lab in vitro experiments. And here I specifically chose those that tie torus knots and links, and in this case torus links. But in recombination experiments, you see many other types of topologies depending on the enzyme and depending on what the substrate looks like. If instead of starting with a non-knot, you start with a link, well, as we saw in a few slides ago, you may unlink instead of unknotting, etc. Okay, so what are site-specific recombinases? They are enzymes. Enzymes are proteins. They bind the DNA. And in this case, for XerCD, you have 4 proteins, 2 copies of C, 2 copies of D. They will come together to form this tetramer. They bind to the 2 recombination sites, which are those 2 short pieces of DNA, about 29 base pairs long, and then they will perform the reconnection in 2 steps. They go through what biologists call a holiday junction intermediate and reconnect. So this is just to give you a glimpse on, well, if we go to a very specific process, a specific example like the one of recombination, well, we can fine-tune the cartoon of reconnection and see more details. And even though this is still a cartoon that doesn't have biochemical details, we see a few more details. If we looked at the reconnection of the fluid vortices, the details would be completely different. But the net effect is that you go from 2 parallel strands on the left right here in horizontal alignment to 2 parallel strands in vertical alignment on the right. So the net effect is the same. How are these enzymes, since they're acting so locally? I said that each site is about 29 base pairs long. 29 base pairs is tiny, tiny, tiny compared to the length of a genome. So how are they inducing topological changes on the DNA at a global level? And one thing we know is that these global topological changes will be a consequence of the geometrical conformation. So again, geometry and topology there, adopted by the DNA substrate prior to recombination. And the cartoon I always show is this one because I think it illustrates simply what I'm trying to say. Here you have a circular DNA molecule on the left with 2 recombination sites in blue and green, and then it's moving around inside the cell or inside the test tube. It's moving around, it's supercoiling, and the 2 sites are brought together by some enzymatic process. If you notice here, if you freeze this frame, there's 3 crossings trapped before recombination happens, whereas at the bottom, there's only 1 crossing trapped before recombination happens. Well, recombination happens right here, so you had 2 sites in parallel alignment. You went to a positive crossing right here, and if we unravel this around, we see that the topology is that of a hop link, a 2-to-1 link. In the bottom, same thing, 2 sites in parallel alignment, introduce a positive crossing, so the local reaction is identical, top and bottom, but now we get an unlink. So the topology of the product really depended on what the geometry was of the conformation, so what the embedding was before the reconnection event. Okay, so we will model local reconnection as a band surgery. And the formal definition of a band surgery is you start with a link in 3-dimensional space, either R3 or S3, and a band will be an embedding of an I cross I, where I is the unit interval, so I cross I is a rectangle, like this blue rectangle right here, into S3, such that the inverse image of the link, of the original link under this embedding is 1 pair of opposite edges in the rectangle. So in this case, top and bottom. And we are going to denote L sub B, the new link that is obtained by replacing these top and bottom edges with the left and right opposite edges in the rectangle. And that is called a band surgery. So it's look at the image. The idea is very simple. The definition can get a bit hard to understand. The orientation of the sites, I mentioned already a few times, is very important. We can have a circle with 2 recombination sites that are either in direct repeat or in inverted repeat. Direct repeat, if you start walking, say here at the top, start walking in a clockwise manner, you hit the first site and you start reading it. You read a word, you keep walking, you hit the second site, and when you read the blue word, it reads exactly the same as a green word. Then the sites are said to be in direct repeat. If when you hit the blue word, after having read the green word, you read the reverse word, then the sites are said to be in inverted repeat. If you perform reconnection between sites in direct repeat, you will always obtain, if the two sites are in a circle, you will always obtain a two-component link. So this process is called a coherent band surgery, and it takes you from a knot to a link and then back to a knot if you do it again. If you do the reconnection between two sites in different components of a link. Conversely, if you do reconnection on two sites that are in inverted repeat, you go from a one-component knot to a one-component knot. And now the orientations are said to be non-coherent, and therefore we consider the substrate and product as being not oriented. We do keep track in our minds of the orientation of the sites just to remember whether we have coherent or non-coherent banding, but in the non-coherent case, the orientation of the molecule itself is not consistent, so we just consider this as the unoriented case, and we consider the top as an oriented case. Okay, our model relies on the Tangle method that was proposed by Ernst and Sumners in the late 1980s. And Ernst and Sumners saw this electron microscopy image of enzymes bound to the DNA. So here you have an enzymatic complex at the top right of the figure and two loops popping out of the complex. These loops represent DNA and the complex represents the enzymes. So they decided to model the complex as a three-dimensional ball. So a ball in three-dimensional space. This ball intersects the substrate into two strings, although it could be three strings. In our case, here we're working with two strings. So it intersects the ball in two strings. So now you have two strings inside and two strings outside. And you know that something is happening inside that complex that changes the topology of the substrate. And as you can see in this image, it will be very, very difficult to actually look with a microscope and see what is happening inside the complex. So we need mathematics to look in. So this picture on the left becomes a mathematical object called the numerator of the tangle E. E represents the enzyme and the bound DNA. And if we look inside the enzyme with the bound DNA, we partition this blue ball into two compartments, O and P. P contains the two recombination sites, the regions of the recombination sites that are going to be cleaved and reconnected. So here P goes to R, and reconnection proceeds by what we call Tangle surgery, where we replace P with R, and every else that is interesting, geometrically interesting or topologically interesting in the complex is pushed outside into the tangle O, and O stands for outside tangle. Okay, so now we'll have the numerator of O plus P, and we can write two tangle equations, one for the substrate. Numerator of O plus P is the substrate and is a link here that I'm calling K naught. And numerator of O plus R is equal to the product, which is another link, K1, here the unlink. And the question is, how do we solve these equations and solve for O, P, and R? Now, solving for O, P, and R requires us to understand something about these tangles, to know what type of tangles these are. And I don't really have time to go into the details of the tangle method, but there's three different families of tangles: rational, locally knotted, and prime tangles. the work of Ernst and Sumner's allows us to solve these equations if we know that the tangles involved are rational. If the tangles involved are not rational, then solving the equations becomes much harder, and there's been work in that direction, including some of our work, Isabel Darcy's work, Dorothy Buck's work, continued work of Klaus Ernst and DeWitt Sumners, etc. And also Koyashi Mukawa, Kai Ishihara, Matt Rathbone, a lot of people throughout the years. Okay, but so how do we first determine what kind of tangle do we have? The main strategy that has been used, and is a very beautiful, elegant method, is to go up to the double branch cyclic covering space, and we take this conformation, which is, remember, your substrate, numerator of O plus P, your product, numerator of O plus R. So this is one link, and this is a different link. Lift them up to the double branch cyclic covering space and look at the tangle surgery upstairs. So if this, for example, was a non-knot, it would lift to all of S3, and you can decompose S3 as the disjoint union of two solid tori. One solid torus can be seen as the neighborhood of a knot, and then the other one is the exterior of that knot. And tangle surgery lifts to a dense surgery that will remove one of those solid tori and reinsert it in a different way to obtain a different space. This space is typically a length space because the knot or the link seen down here is a two-bridge knot in general in the experiments that have been observed. So now you have, in that particular example that I'm walking you through, S3 going, so all of three-dimensional space going into a length space via a process called dense surgery, which is that process of carving out a solid torus and reinserting it in a different fashion. So tangle surgery is equivalent to dense surgery in covering spaces. The key spaces for us are these length spaces, which are unions of solid tori, where the meridian of one of the tori is glued to an interesting curve on the boundary of the other torus, and the slope of that boundary curve determines the name of the length space, LPQ. I won't get into that. And low-dimensional topologies are very interested in studying dense surgery along knots in three manifolds. So this is just putting in words what I said when I walked you through the example, how you can remove one solid torus and re-glue it by changing. So here, for example, you have the meridian in V in red. Originally, it may have been glued to a longitude in M or to this other meridian mu in M, and then in the dense surgery, now you glue it to the curve alpha, which is a different curve on the torus. And a lot of what we do relies on the Montesino trick, which tells you that, well, we know that the branch double cover of a T2N, and why T2N? Because, well, that's what we're interested in based on the recombination experiments. The double branch cover of T2N is a length space LN1, and from Montesino, we know that rational tangle replacement in K corresponds to this rational surgery in the branch double cover. So a band surgery from a torus link to another torus link will lift to an integral dense surgery from a length space to another length space. And here at the bottom, I'm putting two examples. So band surgery from the trefoil to a 2N torus knot or link will lift to a dense surgery from L31 to LN1. And here the question is, well, what kind of ends are allowed in such a process? And with Alison Moore, who was a postdoc here in the last 3 years, and now she's assistant professor at Virginia Commonwealth, and with Ty Littman, who is at North Carolina State, we asked these questions, starting with the trefoil, where can we go by non-coherent and coherent banding? And, well, we answered that question, that these type of bandings can only happen if n is plus or minus 1, 3, or 7, and here's a picture. In the non-coherent case, so not to not, and in the coherent case, if n is plus or minus 2, 4, or minus 6. Some of these results are new, some of them are not new, and their key results are of Isabel Darcy's, but they and Koyashi Mokawa, they all come together into these two pictures. Sorry about that, I don't know what happened. Okay. And when the banding exists, we can exhibit the bandings. So we can tell you how this process is happening. There's well, I haven't checked if this paper has been published, but this appeared in the archive earlier this year. It's a study that builds up on our study, and the tools in our study allowed us to go a bit further than older theoretical works because Ty and Allison came with all this machinery from not-fair homology that allowed us to say more than we could in the past. And some of that machinery was machinery of Wu. And Wu and Yang took that idea and brought it further to give us more answers about obtaining one length space from another one by dense surgery. Okay, it's 8:50. Am I supposed to stop or can I keep going for a little bit? The width.
I'd say keep going.
Okay, I keep going then. Okay, so let me tell you, I always take longer because I explain too much. Okay, let me tell you about the pathways. So remember, we were talking about unlinking DNA links by site-specific recombination. And this is a model that the biologists proposed. This is also the type of pathway that is observed in the experiment done by the physicists in Chicago with fluid vortices. And the best way to show it is with a movie, if it will work. Oh, interesting. Movie's not working. Okay, you don't get a movie. Sorry about that. Okay, so here you would see the Hopf link made of bubbles in a fluid, so a fluid vortex that is tied into a Hopf link. And as it dissipates, you see reconnection right here, which will take the Hopf link into an unknot, and then reconnection right there, which will take the unknot to the unlink. And that movie was working earlier. But so these three last steps in this pathway are observed, and for the trefoil, they also observe similar pathway where you go from trefoil to Hopf link to unknot to unlink, just like for DNA. All right, and so our goal is to understand reconnection events involving T2N torus knots and links, and our motivation was recombination in DNA, but it could also be the reconnection in vortices. You model that reconnection as a band surgery. You look at band surgeries mediating transitions between topological knots or between knots and links, and you go up to the covering spaces and try to say something about the transition between the corresponding length spaces using tools from low-dimensional topology, and more recently with the tools of Littman and Moore using tools from Heeger-Fleur homology. Okay, key question, what are the shortest pathways for this? So we start with a 6-crossing torus link, we go to a non-link. Is this the fastest way to do that? So that's related to Renzo's question. Can you do it quicker and better? And the answer that we have here is that no, you cannot do it faster. If you have a 2-to-n torus link with the orientation of a sites predetermined, so in this case you cannot see the arrows, but the orientation is what we call a parallel orientation of the sites, then the shortest pathway has 6 steps. If instead of having T2,6 you had T2,2n, the shortest pathways would have 2n steps. And okay, so we know the length of the shortest pathway. can we say something more about that? To say something more about that, like is the pathway unique or not, we need to make some assumptions. We go back to the experiments and we make the assumption that the complexity of the product goes down at every step of the reaction. That's suggested by the experiments, and also biologically, you want to unlink. So your end product has to be the unlink, but, well, are you really going down in complexity at every step? We don't know. So let's assume we are. If we assume we are going down in complexity at every step, then this is the only answer. We show mathematically that this is the only possible pathway to take a T2 to N torus link to the unlink. If we relax that assumption, because we don't like such a strong assumption, and instead of saying goes down, the complexity goes down, we say goes down or stays the same, then we do find other pathways, and the other pathways are shown here. We go from the 6 crossing torus link to the unlink, and there's 9 possible pathways, and some of these pathways have been fully characterized by us and by others, and the characterization relies on studying the effect of local reconnection on link invariants such as signature, Jones polynomial, Q polynomial, ARF invariant. And we rely heavily on, when we're looking at signature, on a result by Murasugi that tells you that if L and L prime are related by band surgery, then the difference in the signature is smaller or equal than 1. And this result is a very beautiful result, very powerful. And when Alison and I were working on studying reconnection, we wanted to do reconnection in the case of non-coherent banding, so from a knot to a knot, not only from a knot to a link. Murasugi's result applies from a knot to a link. And so we wanted to ask, is there a way to, can we find a result similar to Murasugi's? Oh, my picture flipped. Well, and we don't have a result as strong as Murasugi's, but we do have a new result that shows that if K and K prime are alternating knots, so now we're doing band surgery between 2 knots, and they have the same determinant, and there should be an equal sign there, for M, a square free integer, then if K and K prime are related by non-coherent band surgery, the difference between the signatures is either 0 or 8 in absolute value. Okay. So the answer to the question we posed is yes, there are other pathways, and we wanted to ask what pathways are more probable. We do that computationally. We model the chains as chains in the computer, and we perturb the chains using Monte Carlo algorithms to find the whole possible space, to sample the space of conformations for each chain, and we perform reconnection between chains, and in that way we're able to go back to this figure and assign weights to this graph. And here we see that even though there's more pathways, the pathway along the diagonal, which was the one originally proposed by the biologists, is the most likely by far. These are probabilities under each arrow, but computationally we put absolutely no assumption. So here I'm showing you the results for only the pathways that go down in complexity at every step or remain the same, but computationally we look at all possible pathways, and this picture is showing you some of them. All pathways, we studied pathways between over 700 different knots and links, and for each one of the edges in this graph, there's millions of iterations in the Monte Carlo simulations, and the thickness of the graph represents the weight of the graph. So what this is showing is the thicker edges represent the transitions with higher probability, and here in red you see the transition with the highest probability in the computer simulation where there's no assumptions at all. And here you see going from the 9-1 torus knot to the 8-1 torus link, 7-1 to 6-1 to 5-1, 4-1, etc. This corresponds to that pathway that was proposed by the biologists. And I will finish here because I'm out of time. I wanted to tell you about chirality. Maybe I'll leave you just with my chirality is important. So that's the one thing that I want to say. Orientation is very important. So chirality is important, and I illustrate that taking the 4-crossing torus link. If you look at the 4-crossing torus link and at the graph of that 4-crossing torus link by reconnection, well, here you see that this 4-crossing torus link goes to the 3 different knots with very different probabilities. The one that the transition with highest probability takes it to the trefoil. But if we take a different chirality of the 4-crossing torus link, and you'll see that in the next image. So here's the one I had before. Here you have a different one. Well, now look, the main product is the unknot, which you didn't have on the left. And the graph is completely different. So we need to be mindful of chirality. We need to have a solid nomenclature, and that takes a lot of time and effort, but it needs to be done. So we redid the knot tables, and I'll leave you with that. And we redid the link tables, and that's the end of my talk. Thank you so much.
Well, Mariel, thank you very much. A terrific talk. How about some questions?
Hello, can you hear me?
Yes, I can hear you.
Can I ask, biological molecules, especially human DNA, is very long and coiled, supercoiled around chromatin and forms loops and bands and so on. And does all of that, there has to be a machinery for dealing with all of that stuff, and does that interact with the topological pathways?
Yes, and we don't know the details. So we know that topoisomerases are probably the main machines to control the topological stability of a genome as long as a human genome. So topoisomerases are there, even though human chromosomes are linear, and in theory you could think, well, if a knot is tied, it will fall off the end. Because human chromosomes are so immensely long, it has been observed that they, and it has been proposed for already maybe 2 or 3 decades, that they self-organize in topological domains, what biologists call topological domains. And in our naive mathematician's mind, we can think of a topological domain as a loop, and this DNA weaving into smaller loops. Each one of those loops could be knotted or could interlink with a neighboring loop. And if that happens, the thought is that topoisomerases go in and unlink and unknot. However, recent studies by the Roca lab and recent studies also by Masa Shora in the FIRE lab in Stanford and in collaboration with Steve Levine show that, that small circles, which could be plasmids, there's some level of knotting or linking in the cells. Is that something that is stable? Is that something that serves a purpose? We don't know. If it's not stable, if these knots and links need always to be removed, then yes, the topoisomerases and maybe the recombinases will be doing that job. Does that answer your question?
Yes, yes. I would think all of that supercoiling just to fit it in small areas is accompanied by an awful lot of writhe. And the writhe might say something about the chirality or whatever.
Yeah, so in the cells, the supercoiling tends to be negative supercoiling. So yes, there is a chirality bias in there. You tend to have negative coiling to go with, well, of course, I mean, inherently DNA is chiral, so it is a right-handed double helix. And in E. coli cells, in bacterial cells, the supercoiling is negative at a level of about -7%. So the writhe is important to study the writhe of those molecules. Yeah.
So some more questions. Okay, I've got one. So Mariel, you had this chord diagram. You had, lots and lots of pathways. So what was the upper bound on complexity for those diagrams? Number of
You
crossings.
mean number of crossings? Okay, so
Yeah.
if I showed you the actual diagram, you would just see like a solid green here because there's so many edges. We started with about 430, I think, a little bit under 450 different knots and links as the substrates. We did a computational experiment for each one of them. So we embedded them in the simple cubic lattice. We used BFA-CF algorithm to perturb and sample the space of conformations. We were very careful with the statistics. We were very careful to do uniform sampling and to have a statistically significant sample for each one of those 450 different knots and links. And once we had a good set of samples for each topology, we looked for recombination locations. Each one of the topologies has maybe none, maybe one, maybe two, maybe five recombination locations. If there was more than one, we picked one at random, uniformly at random, and performed recombination and computed the resulting topology. So if we started with a link, we computed the knot that was obtained by reconnection. And then in that way, we informed the weights in this graph. We informed the probabilities of going from, for example, the trefoil to the Hopf link by looking at how many times the trefoil went to the Hopf link in our trefoil experiment. And we repeated that for each one of those 450 topologies. The products, of course, could be much more complicated. In our data set, I think there's about 910 different products. There's others that are unknown, where the Hopf polynomial just spits out something that we don't recognize. But I mean, identification algorithms are not great. So those we excluded from the study. And so this graph, the outside of the graph are the nodes of the graph. So these are the vertices. On the left, you have the links, and on the right, you have the knots. And all in all, in the real graph, there's over 900 vertices. And I don't know the number of edges. We didn't count them, but there's a lot of edges.
Okay.
I mean, not quite 2 to the 900, but there's a lot of edges.
So any other questions? Okay, well, Mariel, thank you very much. We really enjoyed it. Thank you.
Thank you. Thank you, everyone.